Hello everybody, we are studying about biotechnology principle and processes. Today we are going to study about an important tools of recombinant DNA, that is restriction enzyme. So what is restriction enzyme? How is it invented? Let's talk about it. ब्याक्टेरियालाई जब भाइरसले इन्फेक्ट गर्छ कुरा बुझ्नु मलाई when bacteria infect the virus हैन भनेपछि we know that virus that infect bacteria लाई bacteriophage भन्छ भनेर we have already studied in the molecular biology. So okay, जब ब्याक्टेरियालाई चाहिँ भाइरसले इन्फेक्ट गर्छ त्यति बेला भाइरसले it used to insert its DNA inside the body of bacteria, isn't it? र enzyme which is present in the body of bacteria, enzyme which is present in the body of bacteria, त्यो एन्जाइमले चाहिँ के गर्छ भन्दा it used to cut the DNA of virus. भनेपछि enzyme which is present in the body of bacteria will cut the DNA of the virus so that DNA will not able to replicate and bacteria will protect itself from the infection of the virus. र that enzyme is called as a restriction enzyme. त्यही एन्जाइमलाई चाहिँ हामी के भन्छौँ restriction enzyme भनेर भन्छौँ. restriction enzyme भनेको चाहिँ के रहेछ? enzyme which is present in the body of bacteria which used to cut the DNA of the virus and protect themselves from the infection of the virus. त्यसलाई नै हामी के भन्छौँ? restriction enzyme भनेर भन्छौँ. Is that clear? So okay, in 1963, अब 1963 मा in the body of E. coli को body मा, in the body of E. coli, two types of enzymes were found. One is methylated and the other is endonucleases. So this restriction and enzyme is also called as a restriction endonucleases पनि भनिन्छ, and it is also called as a molecular scissor पनि भनिन्छ. It is also called as a molecular scissor. So restriction enzyme is also called as a restriction endonucleases and it is also called molecular scissor. किन भनिन्छ भन्दाखेरि because it used to cut the sequence of the DNA. Yes or no? Okay. र in 1963 in the body of E. coli, two types of enzymes were found, methylated and endonucleases. र E. coli को आफ्नो उयो पनि छ, DNA छ. So whenever E. coli is infected by virus, yes or no? Whenever E. coli is infected by virus, that means virus will insert its DNA. Yes or no? र whenever DNA of the virus is inserted inside the body of E. coli, this endonucleases will cut this DNA. यो DNA लाई काटिदिन्छ so that this DNA will not able to multiply. तर अर्को कुरा के हुन्छ भन्दाखेरि this endonucleases will not able to recognize self and the non self भन्नु मतलब foreign DNA र आफ्नो DNA लाई उले के गर्न सक्दैन, recognize गर्न सक्दैन. That's why it will also cut the sequence of the जुन चाहिँ सिक्वेन्समा यहाँनिर काटेको हुन्छ त्यही सिक्वेन्समा यहाँनिर आएर पनि काटिदिन्छ. भनेपछि आफ्नो DNA नै उले काटिदिएको रहेछ. भनेपछि अब this DNA should be protected. So to protect this own DNA, this methylated enzyme will add the methyl group. This methylated enzyme will add the methyl group. Is that clear? So this is the functions of two enzymes which are present in the body of E. coli. What are the functions of two enzymes? What are the functions of the two enzymes of the E. coli भन्दाखेरि one used to add the methyl group, एउटाले methyl group add गर्दैछ भने अर्कोले it used to cut the DNA. एउटाले चाहिँ DNA लाई काट्ने काम गर्थ्यो भने अर्को एन्जाइम्सले चाहिँ methyl group add गरेर it used to protect its own DNA. Okay, this is about and it was happened in which year? It was happened in the year of 1963. 1963 मा चाहिँ यो भएको थियो. Is that clear? Okay, and we know that virus that infect bacteria लाई हामी के भन्छौँ रे? Bacteriophage भनेर भन्छौँ. कुरा बुझ्नु होला नि? Okay, अब this is about. Now next one. Okay. First restriction enzyme. Let's talk about this. First restriction enzyme. First restriction enzyme चाहिँ कुन चाहिँ हो भन्दाखेरि HIND2 हो. The first restriction enzyme is HIND2. Is that clear? And it used to cut at a specific region. यसले स्पेसिफिक रिजनमा काट्ने गर्दछ. It used to cut the specific region, and what is that specific region? About that we'll talk later on. Okay. अब अर्को कुरा गरौँ. What is restriction and enzymes? Restriction enzyme भनेको के हो भन्दाखेरि it is the large group of enzyme called nucleases. It is a large group of enzyme. What is restriction enzyme? It is the large group of enzymes called nucleases. So there are two types of nucleases. Number one is exonucleases and number two is endonucleases. So there are two types of nucleases, exonucleases, endonucleases. Exonucleases will cut at one end of DNA. यसले one end of DNA मा काट्छ भने it used to cut at a specific region inside DNA. भनेपछि यसले एउटा स्पेसिफिक रिजनमा DNA को भित्रपट्टि भएको कुनै एउटा स्पेसिफिक रिजनमा चाहिँ यसले काट्ने गर्दछ. So these are the two types of nucleases. So what is restriction enzymes? It is the large group of enzyme called nucleases. There are two types of nucleases, endonucleases and exonucleases. Exonucleases ले one end मा कट गर्छ भने endonucleases ले एउटा स्पेसिफिक रिजनमा कट गर्छ. So what is that specific reason? Let's talk about it. Okay. So restriction enzyme or restriction endonuclease, it used to cut at a specific region, that is palindromic nucleotide sequence. भनेपछि restriction endonuclease ले कुनै एउटा स्पेसिफिक रिजनमा काट्छ र त्यो स्पेसिफिक रिजनलाई चाहिँ हामी के भन्छौँ? palindromic nucleotide sequence. अब palindromic nucleotide sequence भनेको के हो भन्दाखेरि we know that DNA को सर्कुलर DNA होस् या लिनियर DNA होस् हैन. अब यो सर्कुलर DNA छ, यो तिम्रो लिनियर DNA हैन. र अब एन्जाइमले पहिला के गर्छ? एउटा स्पेसिफिक रिजनमा यसरी सपोज एन्जाइम बाइन्ड भइसक्यो, स्पेसिफिक रिजनमा गएर उसले त्यो स्पेसिफिक रिजनलाई जब उसले पत्ता लगाइसकेपछि what happens भन्दाखेरि त्यो अब कुनै पनि स्पेसिफिक रिजनमा जब उसले पत्ता लगाइसकेपछि त्यो जग्गामा उसले त्यो सिक्वेन्सलाई उसले काट्छ र त्यो स्पेसिफिक रिजन चाहिँ के हो? palindromic nucleotide sequence भनेको चाहिँ त्यो सिक्वेन्स हो जहाँनिर चाहिँ द वर्ड चाहिँ दुईपट्टिबाटै एउटै मलायालम एउटै प्रकारले दुईटैपट्टिबाट रिड गर्ने लेटरलाई हामी palindromic nucleotide sequence भनेर भन्छौँ. जस्तो प्रकारले मलायालम यताबाट मलायालम, is not it from both sides, is not it? अब यो DNA भित्र भएको त्यो palindromic nucleotide sequence चाहिँ के हो भन्दाखेरि that is GAATTC. यताबाट पनि GATTC भने यताबाट पनि GAATTC. So this sequence is called as a palindrome nucleotide sequence. So restriction enzyme used to cut the palindromic nucleotide sequence. So in this type of base pair मा चाहिँ गएर चाहिँ के गर्छ? restriction endonucleases ले या restriction enzyme ले चाहिँ के गर्दछ? क्लिप गर्छ. Is that clear? So this is about the restriction enzyme. So in the next video we'll learn about the how is that foreign DNA is isolated. About that we'll learn in the next video. आजलाई यति, फेरि भेटौँला. Thank you very much.